Welcome and thank you for joining today's presentation on apoptosis research. Our focus will be on understanding the mechanisms behind programmed cell death, its roles in health and disease, and the methods scientists use to study it. Let's dive in to see why apoptosis is more than just cell death. It's an essential process that keeps cellular life in balance. To start, apoptosis, or programmed cell death, is a precisely regulated process where cells essentially self-destruct to benefit the overall health of an organism. It's critical in development, immune response, and maintaining tissue balance. Unlike uncontrolled cell death or necrosis, apoptosis is orderly and avoids harming neighboring cells. This self-regulation is key to health, and when it's disrupted, diseases can develop. So, apoptosis is the ultimate act of cellular responsibility. Cells remove themselves before they can cause harm. Apoptosis operates through two main pathways, the intrinsic or mitochondrial pathway and the extrinsic pathway, each triggered by different signals. In the intrinsic pathway, internal stress like DNA damage signals BCL2 family proteins, leading to the release of cytochrome C from mitochondria and activation of caspases that initiate cell dismantling. The extrinsic pathway is externally triggered when death ligands, such as FA's ligand or TNF, bind to receptors on the cell's surface, also leading to caspase activation. These cascades ensure the cell follows a controlled sequence toward a clean death. When apoptosis malfunctions, it plays a central role in many diseases. In cancer, for example, cells evade apoptosis, allowing unchecked growth and resistance to standard therapies. Researchers are targeting this resistance by developing therapies like BCL2 inhibitors to reactivate apoptosis in tumors. Meanwhile, in neurodegenerative diseases such as Alzheimer's and Parkinson's, apoptosis can become overactive, causing excessive neuron death. Modulating apoptosis could help protect neurons and slow disease progression, making this field critical in advancing both cancer and neurodegeneration research. Studying apoptosis requires precision, and we have a range of methods to detect and measure it. Flow cytometry with NX&V and propydium iodide staining is widely used for detecting early and late apoptosis. The Tunel assay identifies DNA fragmentation, a hallmark of apoptosis, while Western blotting and ELISA allow us to quantify specific apoptosis proteins like caspase-3. For visual insight, microscopy techniques, especially fluorescence and live cell imaging, enable us to see the progression of apoptosis in real time, helping researchers capture this process as it unfolds. To investigate apoptosis, researchers rely on various models. In vitro models like engineered cell lines can be induced to undergo apoptosis, allowing detailed molecular study. In vivo, genetically modified mouse models, such as BCL2 knockouts, allow us to observe apoptosis at the organismal level. Recently, organoids and 3D cell cultures have emerged as valuable tools, mimicking tissue environments more closely and providing new ways to study apoptosis in a more realistic context. Each model adds a layer of understanding to this complex process. Apoptosis research has evolved significantly. In 1842, Carl Vogt made the first observations of natural cell death. In 1792, John F.R. Care Andrew H. Willie and Alistair Curry formally introduced the term apoptosis, defining it as a structured, orderly process. Key breakthroughs came in the 1980s and 90s, with discoveries of caspases and BCL2 proteins, and by the 2000s, apoptosis was recognized as a therapeutic target, especially in cancer. These milestones have transformed apoptosis from an observed phenomenon to a core focus in medical research. Apoptosis research continues to advance, with emerging applications in fields like cancer immunotherapy and neurodegenerative diseases. Scientists are now working to develop therapies that selectively trigger or inhibit apoptosis, depending on the disease context. Artificial intelligence is also entering the field, helping researchers process and analyze large data sets to identify new patterns in cell death regulation. These innovations promise new insights and therapies that could reshape how we approach diseases driven by apoptosis dysregulation. In conclusion, apoptosis is more than a process. It's a finely tuned system crucial for life, balancing cell renewal and death. Its role in diseases like cancer and neurodegeneration has made apoptosis research a cornerstone of medical science, guiding the development of targeted therapies. Understanding its pathways, detecting its markers, and applying innovative research methods are helping scientists unlock new ways to manage health and disease. Thank you for watching. Let us know what other topics would you like us to cover in our next videos.